With the preliminary words and concepts now under our belts from the previous section, we can begin to discuss experimental design. A scientific experiment is when researchers will add or remove certain factors into a system in order to see what occurs as a result. In doing so, they'll measure how these manipulations change the behavior and outcomes of a system. A system here can be defined as any set of interrelated and interacting parts that together fulfill a certain function. It's a broad definition that can be applied to almost anything, whether it's a single atom or an organism or an ecosystem, or even the universe as a whole. Scientific experimentation is perhaps the most important tool that separates modern science from its more philosophy and observation-based predecessors. When we combine these experiments with quantitative analysis, a fancy phrase for using number-based measurements like height, mass, speed, etc., we really have all the tools required to do science as it's currently known. The rest is just details. Let's then talk about scientific studies. When we think of the word study or studies, it normally evokes images of us reading or listening to content that's already been worked out and produced by others, such as in the case of studying for an exam. But like many words in the world of science, it takes on a slightly different meaning here. We often use the phrase doing a scientific study, which means attempting to discover something new that no one else has worked out before. However, we'll see later that sometimes doing a study can also mean reconfirming the discoveries of others. This may also be confused at times, since in most schools, colleges, and universities, students are still tasked with studying science in the more colloquial sense so as to pass their exams. That aside, there are generally speaking two different methods of performing a study in science. The first is called an observational study. As the name suggests, this is where you as a researcher would observe a system and not make any alterations to it. Instead, you would only take measurements. An example would be if you decided to see if there was a difference in the sizes of Asian elephants who eat primarily tree leaves and those who eat primarily shrub leaves. In this case, you might simply observe two different wild populations of these elephants and take size and weight measurements for them. This kind of study is contrasted by an experimental study. This is where you make an observation and then manipulate a factor or set of factors within that system to see if it changes anything. An example, you walk by a cabbage patch and find that the plants are turning yellow or brown, indicating that they're probably not as healthy as would be ideal. You predict that the addition of fertilizer would improve their condition, and so you go about applying that fertilizer to the patch in an attempt to prove or disprove that. Note that in both of these scenarios, we're starting with an observation. The difference is that with the elephant study, we're only observing and measuring. In the cabbage scenario, we're observing, measuring, and manipulating something. Maybe the most fundamental tool that scientists employ is the scientific method. Put succinctly, this is a set of steps they're meant to follow if they want to test the validity of their ideas. Let's illustrate this by drawing on our cabbage patch example from the previous slide. We start by making an observation. In this case, that the cabbages look sickly. After making this observation, we formulate a question about what's happening. In this case, why do they look sick? Then we come up with a tentative answer to the question, which we'll call a hypothesis. This word connects to another word that's maybe more familiar to most of us, hypothetical. This makes sense because the hypothesis quite literally acts as a hypothetical answer to your question. It comes down to us from the Greek hypo, meaning under, and thesis, meaning placement or proposition. So literally, it's a proposition that's placed under a scientific investigation, meant to serve as a guiding foundation of sorts. A common convention in scientific philosophy is that a hypothesis should have as few arbitrary assumptions as possible. This would be assumptions that we say are non-falsifiable, such as a variation on our hypothesis that the soil nutrients are lacking because some other creature is siphoning them away. While that may be the case, at this time it would only serve to complicate our current hypothesis. This is also a large part of why certain so-called supernatural hypotheses are often considered non-scientific. It's not necessarily because they can't be true, but because they rely on assumptions that can't be tested using the scientific method. Anyway, from the hypothesis, we're then going to make a prediction, which means say before. This is usually formulated as an if-then statement. In our case, if fertilizer is added to the soil, then the health of the cabbages will improve. The next step is to create and perform an experiment that serves to test the validity of your hypothesis and prediction, followed by collecting and analyzing your data, or raw mathematical measurements. We'll be talking about how to run experiments and data analysis in greater detail a little later on in this lesson, but for now, we can say that one of two things will occur. Either your hypothesis and prediction are supported, in which case you will want to communicate your findings to other scientists or interested parties. You then might be inclined to continue the research by coming up with a follow-up hypothesis and prediction. Maybe we could start looking into which specific nutrients the soil is lacking. Alternatively, your hypothesis and prediction might be unsupported. In these cases, it's still useful to communicate your findings so that others aren't as inclined to test your failed hypothesis again, though sometimes retesting a hypothesis can actually still be useful. From there, you can either give up and do something else with your time, or if you're feeling tenacious, you can test another hypothesis. Maybe the soil nutrition is fine, but there are some parasites or other pests present causing the crops to fail. It should be noted as well that the scientific method has gone through many changes throughout history, and there are some slight variations of it depending on the field that you're in or the minutiae of the questions being asked. Also, you might find elsewhere similar flowcharts that have additional steps in them. These are, for the most part, it seems perfectly valid, but I kept it here to the steps that I felt were essential to mention at this point. Another important concept in science is that of falsifiability, or otherwise called refutability. For a fact, law, or theory to be considered scientific, it must be possible to show that it's wrong, even if it's true. Another way of saying this is that they must be falsifiable or refutable. Although the idea has been around for longer, the term itself was first coined by philosopher of science Karl Popper in his 1934 book, The Logic of Scientific Discovery. There's an often cited example when falsifiability is being taught in the form of the statement, "all swans are white." In order for this statement to be proven, all of the swans on the planet would have to be observed to be white. Since this is an impossible task, these criteria cannot be met. In addition, observing just one single black swan would immediately falsify the statement. Thus, statements such as opinions, such as blueberries taste better than raspberries, or speculations, such as there are invisible, undetectable dragons flying around in the sky, are non-falsifiable as well. They cannot be proven true or false. The concept simply can't apply. Note that non-falsifiable statements may indeed also be true. It doesn't mean that they are false statements. No one is saying that they have no validity towards describing reality, only that they cannot be used as a basis for scientific inquiry. The tenet of falsifiability implies that there's no such thing as settled science. To claim that any science is settled is one of the most anti-scientific claims that can be made. And it's also non-falsifiable in itself, so how about that? A proper scientist then sets out to prove their hypotheses wrong, not right. Once you have done everything in your power to disprove your ideas and failed to do so, then you can logically conclude that it's an accurate model of reality. As with everything, this is a loaded discussion with many takes by many thinkers. It's even being argued now by some that falsification as a means to gather knowledge is now a defunct way of thinking. However, this is currently a fringe position, and thus perhaps a discussion for a future module. Now, let's expand on our understanding of an experiment by returning to our sick cabbages. The original hypothesis we came up with was that the cabbages were malnourished because the soil lacked nutrients, and figured a solution lied in adding fertilizer to the soil. Well, let's run that experiment. Here's our field and our cabbages. It might be assumed that if we were to test our hypothesis, that we'd apply fertilizer to the entire cabbage patch first and then watch to see what changes. But if we were to do this, during the waiting period to see if the cabbages improve, what if something else were to take place that could affect their development? What if it were to rain? Or what if the cabbages were able to figure out a way to heal themselves without the need of some external intervention? Realizations like this are why certain precautions are required to run an experiment properly. So we're going to first designate two treatment groups and place each cabbage into one of these two groups. For simplicity, I've cut the garden here in half and am designating the two groups in this way, though in real life you wouldn't necessarily need to do it this way, and there may even be some reasons not to. The first treatment we call the experimental group. These are the cabbages that will be getting the fertilizer. The second we call the control group, which will be left alone and not receive fertilizer. The rationale behind these names is that for the control group, we're controlling for extraneous factors that might otherwise mess up our data. since every single cabbage in this plot, regardless of which of the two groups it's in, will be receiving the same amount of water and sunlight, growing in the same soil, and under threat of the same pests and pathogens. The only thing that's different between them is that one has fertilizer and the other one doesn't. Now, we add the fertilizer to the experimental group. Mind you, this has to be a precise amount added to each one. We can't just eyeball it in science. It also has to all be added at the same time, or as close to the same time as possible. If we don't do this, then there might be differences between the cabbages just within the experimental group that were the result of longer exposure to the fertilizer. After this, we wait a predetermined amount of time, let's say 1 week, and then we return to the cabbage patch to check our results. If the cabbages in the experimental group tend to look healthier than the ones in the control group, then we can conclude with some confidence that the fertilizer is indeed what was missing, and we can probably go ahead and give some fertilizer to the control group to help them out. If, on the other hand, there is no difference between the two groups, then we'd reject our original hypothesis and conclude that fertilizer probably isn't the factor that's causing sickness. We could give up, or we could come up with a new hypothesis to test. Or maybe we could even test the same hypothesis again, making slight tweaks to the experiment, like adding higher or lower amounts of fertilizer, or maybe even a different type of fertilizer. There's also the possibility that the experimental group actually comes out looking worse than the control group, in which case we might be led to conclude that the soil has too much of some type of nutrient and is causing fertilizer burn, which we just exacerbated by adding even more. When a scientist carries out an experiment and comes up with the results that they'd like to communicate to others, they typically write it up in the form of a scientific paper and submit it to a publishing journal. These journals are similar to periodical magazines in that they're released at regular intervals throughout the year. There are a great many journals out there. Some are more specialized and only accept papers from very particular subfields, such as aquatic botany, which only deals with research papers related to plants and algae that grow in and around water, and brain hemorrhages, which deals, of course, with studies related to, what else, brain hemorrhages. Others might be a bit broader as far as what they'll accept. As a result, though, it's usually more difficult to get your work published there. Examples might include Genetics and the American Sociological Review. Or they can be extremely broad, such as the very well-known Science and Nature, which will accept papers from any scientific field. In general, the broader the journal, the harder it is to get into. And so the slots in Science and Nature are typically reserved for research that's considered absolutely groundbreaking and cutting edge. However, before a scientific paper is published, it must be critiqued or reviewed by a panel of relevant experts, or peers, who work for the journal. Their job, ideally, is to check everything from formatting and spelling to the methodologies employed by the researchers to the interpretations of their collected data. If it passes their scrutiny, it gets published. And if it's not up to snuff, it gets rejected and is then sent back to the researchers for revision. On paper, this sounds like a fantastic idea since it provides a way for scientists to check each other's work and point out flaws, and very often it does serve this purpose. Unfortunately, though, this whole process is now ripe with flaws, which we'll be getting into later on in this module. When we talk about a scientific article, we're talking about something very specific. These aren't the same thing as writing a newspaper editorial or an online blog post or Facebook comment or anything of that nature. There's a strict formatting structure that they're meant to follow. This structure can vary a bit depending on which journal they decide to publish in, but these details are minor. It's more or less the same across the board. The first part is pretty self-explanatory. Your article is going to need a title. However, it's a convention of scientific literature that the title precisely state what the article is about specifically, or the main conclusion of your research. Oftentimes, this means the titles aren't going to be super catchy or concise. It's one of the few genres of literature where this is the case. The first actual section of the scientific paper is going to be an introduction, which does exactly what it sounds like. It introduces the topic that you're about to talk about. Let's use the Cabbage Patch example again. When we write up our report on our experiment, we're going to want to begin the introduction by reviewing research that had already been done prior, which informs our hypothesis and prediction. For example, scientists who had researched the effects of soil malnutrition on other plants, the basic nutrient requirement of cabbages, etc. This would mean citing many articles published previously by other researchers. Introductions tend to start off broad and get more specific as they near the end, concluding with a statement of the hypotheses that you are going to be testing. The second section of a paper is normally the methods or methodology section, which describes in specific detail what was done to run the experiment. In our case, we would talk about the physical materials we used, perhaps scales to measure the mass of the cabbages, the type of fertilizer we used, etc. We'd also explain here how we separated the patch into the experimental and control groups, how much fertilizer we used, when it was applied, what data we collected, and when we decided to do so, as well as any statistical analyses used to determine whether or not the data supported our hypothesis. Next comes the results. This is usually the shortest section of the paper, where authors simply report what happened, what was observed, and what the results of the statistical analyses were. Nothing more, nothing less. No interpretation required. That's saved for the next section, which is called the discussion or conclusion. As you might have guessed, this is where the paper comes to an end. The purpose here is to more thoroughly analyze your results, answering the questions of what they mean and how they can be applied to solving real-world problems. You can also include things like how your experiment is relevant to other areas of research, as well as make suggestions for what types of follow-up questions and experiments could be run to further expand on your discoveries. But just because the paper itself is ended doesn't mean that we're necessarily done. We're also going to need a works cited section. This is a necessary part of the paper that's tacked on at the end that lists out sources that we've referenced throughout the paper, mostly in the introduction and discussion sections, so that the readers can be better assured you weren't just making things up, and they can check the sources of information you're drawing upon. It also serves to provide readers with more content that they can follow up on if your topic was of interest to them. But wait, there's one more. After all is said and done, most articles include what's called an abstract or a summary that they tack on at the very beginning before the introduction section. This serves the same purpose as a TLDR epithet does with posts on the internet. It's a super shortened version of the paper that provides just the main highlights of each of the 4 main sections. This can give other researchers an idea of whether or not your paper is relevant for them to read or cite. But then we get to the famous peer review process. Before a scientific paper is published, it has to be critiqued or reviewed by a panel of relevant experts, or peers, who work for the journal. Their job, ideally, is to check everything from formatting and spelling to methodologies employed by the researchers to the interpretations of the collected data. If it passes their scrutiny, it gets published. If it's not up to snuff, it gets rejected and then is sent back to the researcher or researchers for revision. On paper, this sounds like a fantastic idea since it provides a way for scientists to check each other's work and point out flaws. And very often, it does serve this purpose. Unfortunately, though, this whole process is now ripe with flaws, which we'll be getting into later on in this module. But the process goes like this. First, you're going to write and submit an article to a journal. Next, a review board is going to critique your paper for formatting, methodology, and conclusions. And then one of 2 things is going to happen. The first scenario is that the review board is going to approve your article, and then the article is going to be published in the next edition of their journal. The second option, though, is that they reject your article, in which case the article is going to be sent back to you, and then the authors revise the paper in accordance with the editor's remarks, and then they submit again. Hopefully, after the second or third submission, your paper will be accepted and you can celebrate. But oftentimes there's a bit of an obstacle to even doing your studies in the first place. Scientific research can be expensive. Supplies cost money, and the instruments required for certain fields can run into the thousands, tens of thousands, or sometimes even millions of dollars. This is one of the main reasons why scientific research has historically been the domain of the upper classes of society, others being the fact that they had access to the educational opportunities to afford them the know-how in the first place, and they also didn't have to worry about spending their precious daylight hours earning a living. They could just devote it to research. Most modern scientists, however, can't afford to fund their own research, especially when they're new in their careers. So how is this solved? One of the primary ways is to work at a college or university and receive some funding from the school that you're a part of. Generally, income is brought in through students' tuition payments. Much of this can be allocated to labs so that they can afford their supplies. However, this doesn't always cover everything. Another bread and butter tactic is to write grant proposals to government agencies or organizations who have vested interests in funding scientific research. An example might be a biotechnology company who works in genetics wanting to see research published that helps them to grow their own knowledge base, which can potentially result in the development of new technologies, which then increase their profits. Many researchers nowadays actually spend a good bulk of their time writing these grants, and I've even heard some of them lament that it feels like they spend most of their time doing so, rather than the actual research that they so love. If you work for a private institution, such as a drug company or other biotechnology firm, the funding for research comes in as part of their revenue. So working for them is a good way to avoid writing up grant proposals. You've just got to make sure that you enjoy the work that you're doing and that the fruits of your labor are being put towards ends that you see as ethically appropriate. Within any academic field, not just science, it's of paramount importance that when you're presenting arguments or findings, any claims you make leading up to your conclusion should be cited. This means that you tell your readers and listeners where you read those claims from. For example, if you've been studying why a certain bird population migrates to a particular town every year, it might be necessary to cite a paper written by someone else who first documented what and where that town is and where the birds originally came from. Even outside of academia, citation is just ethical. If someone else influenced your ideas, you should pay homage to them, even if you don't necessarily like that person or fully agree with everything they say. Just as an aside. Proper citation does a good job of keeping us humble in the pursuit of knowledge by reminding us that we owe the majority of that knowledge to others who came before us. There's the old dictum that was first penned by Isaac Newton, though he himself was not the originator of the idea, that went, "If I have seen farther, it is by standing on the shoulders of giants." And on a more pragmatic level, it helps you to avoid accusations of plagiarism, which you definitely don't want. Anyway, that about concludes section 3. I will see you all in the next one.